Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast, where we bring you conversations with leading experts in the GPCR field. Today's episode is extra special. Not only do we get to speak with Dr. Maria Majellaro, co-founder and chief scientific officer at Celtarys Research, but we're also excited to share that we are partnering with Celtarys Research. Celtarys is redefining how we develop and apply fluorescent ligands in GPCR research. Whether you're in drug discovery, pharmacology, or assay development, this collaboration is all about making tools more accessible, more customizable, and more effective. Thank you for joining us on this exciting journey. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and welcome to the Dr. GPCR podcast. Today, I'm very excited to have with me Dr. Maria Majellaro. She is the CSO of a small company in Spain called Celtarys. Today, we're going to learn a little bit more about her career track that led her to where she is today. And I'm very excited to also tell you that Celtarys has become Dr. GPCR's partner. So we're excited to have the Celtarys family within the ecosystem. And today we're going to get to know a little bit more about Maria and Celtarys. Maria, welcome to the podcast.
Thank you so much. And thank you so much for the invitation. I'm very pleased to be here today.
I'm very excited to have you on the podcast. I know that we've met a few years back, very casually, and I don't even remember how it happened. I think it was through LinkedIn. I don't know if you do remember, but it's been a while since.
It was in the GPCR community. It was, I think, Glisten cost action that then was, the Arnes cost action. So it was in the frame of this. Yes. Mm-hmm.
It's a small world, the GPCR world. So
yeah, the GPCR world.
Very excited to have you on the podcast, Maria. So let's start at the beginning. Could you please introduce yourself, tell us who you are, and what is it that you do?
Okay, so as you mentioned before, I'm the Chief Scientific Officer of Celtarys Research, which is a Spanish startup company, a spin-off of the University of Santiago de Compostela. We define us as a biotech company, but, biotech can mean a lot of things. So basically, we are medicinal chemists. We have been working a lot in the development of modulators. Well, within the group, in the academic group in which I did my postdoc, GPCR modulators. And so basically, we developed this technology that we are applying now for the development of fluorescent probes. That can be used for the development of new assays to support and foster drug discovery programs. So we are basically applying our knowledge about how we can modify these scaffolds to try to keep their activity, affinity, or their biological behavior as similar as possible to the parent compound. And I can say that we are doing quite well. So I'm very happy about that for the moment. But there are always new challenges because, we started with GPCRs, but we are now working with a lot of new targets. And every time is a new world, and we learn every time a lot of new things.
No, I agree with you. And I think, I've worked now a couple of years on small molecules, and it's a challenge because and it's a fun challenge to know, okay, which residue, which group, how do I change the molecule, not only to drive SAR and typical drug discovery, but also how do you label that molecule in order to make sure that, it still keeps its activity. And I think that can be tricky and fun at the same time.
Yeah. Also because, you add to new projects what we have been learning before, because it's not just the ligand, so how you modify this ligand, but also linkers and fluorophores you use. So that there are some fluorophores that are better for a specific application, or linkers that are better for a kind of target, but also a specific type of assay. And so because with our technology, we can really explore different linkers. So just change the reagent we use, we can modify a position of the linker. And so instead, of conventional chemistry in which you have to build the linker with conventional chemistry, so you have to perform one synthetic route for that linker and then another synthetic route for that other linker. In our case, just changing one reagent that sometimes is also commercially available, we can have access to different linkers. And this is why we have been learning a lot also about linkers to be employed. Because you have also these two aspects, pharmacodynamic and pharmacokinetic.
Yeah.
So, it's not a full body, but we can, compare it also in a small assay. So we can refer to the mechanism of action and to the physicochemical properties. So you can have a probe that could be great, but if you are not able to solubilize it, it's not useful. And so we can play with this also with linkers, fluorophores, and modifications.
That's wonderful. When you think about it, you are writing the playbook of understanding, depending on where do you put your linker, does that molecule, the ADME properties of the molecule get affected or not? I think it's wonderful. So tell me, how did you become a scientist? Did you always know as a child that you wanted to become a scientist?
Good question. No, well, I have to say that the main reason why I love science is due to my father, because my father, he's a doctor, he's retired now, but he was like this kind of passion doctor that, want to help people and make everything to help people, not only, through medicines, but also from, an emotional point of view. And I always observed him. I have this image in my mind of when I was a child. And so there was this guy, he fell down because he was feeling bad, and my father, helping him. And so I had this very nice example in my life. I have to say that he always showed us this Esplorando il Corpo Umano is the name in Italy, but it's like a cartoon series. I think it's like Once Upon a Life in English. Okay. And so you have these cartoons and they explain you how the body works. And so you have this kind of macrophages, like Pac-Man style machines that eat pathogens, or like this red people with this backpack and transparent balls.
I think there's a US or international version of this. I think it's called the Magic School Bus. Where the kids get on the bus with the teacher and then they go explore the body. And growing up, I watched it as well.
No, I don't know if it's exactly the same, but I think it's a very funny and creative way to, approach children to science. But I also knew that I could not be a doctor because I'm quite sensitive to, blood. I'm not. I've not issues with injections, but when you are in a first aid, you can see everything, and also the thing that you have to take these decisions, under pressure. You have the life of people on the line. It's. I think I could do it, but I don't know if I could be able to manage it from an emotional point of view, because you don't have to keep it, bring it home.
Yeah,
you have to be able to switch things. My father was great in this, but you have also to know what you can do, and also, I really loved cooking. So we can say that when you want to do a cake or something like that, you have to search for the best recipe. So literature search, basically.
Yes, I agree.
Also interviewing relatives to ask them or friends if they have a good recipe for you. You have to find the best reagents, precursors for the cake, and then you have to test the different conditions. So indeed, for instance, in the last period, I have been optimizing my protocol for a focaccia. That is like this is a typical food from my hometown in south of Italy. It's, we can say, like a pizza. I hope nobody from Bari, my hometown, is listening to me because they could be upset. It's not actually a pizza. It's like a leavened dough
Yes. I think, I think most people know what a focaccia is, and it's delicious when it's well done.
Yeah. And so it's very simple. I mean, you don't have crazy ingredients to use,
Yeah.
but these are the things that really need to be done very well. So I've been optimizing there the order of ingredients, addition, time of levitation, time, the temperature, when do I add the salt, before, after, potato, no potato.
Yeah.
And now I
I
have my
I completely understand that. And it's funny because you brought up the example of baking a cake and having the right recipe and the right references and the right ingredients. And whenever I speak about science to anyone who's not versed in science, the baking the cake is my example, my go-to example as well.
yeah.
Because it's so simple and everybody can relate to cake. Focaccia, you took it to another whole level. I think
Yeah.
that's
No, also because now I have my recipe, and every time people ask me, I share it. So completely open science from that point of view.
well, maybe we can put it on the website under the description of the episode. Do you have it somewhere on a site where you can share a link with us, or just the whole recipe? I think it could be a really fun thing.
Yeah. I can. Would be a pleasure.
Thank you. No, I think that's wonderful. So you mentioned, coming from a small town in the south of Italy, growing up as a child, having your dad as, the figure for helping people, but also realizing that medicine was not for you. By the way, you're not the only one. I think there's a lot of us who did a PhD who then very quickly realized that medicine is not for us. When you went to school, what were some of your favorite, favorite topics, subjects that you were studying? Because obviously we were talking about chemistry and linkers, but where does biology and the chemistry meet in your career?
Yeah, actually, I mean, because in high school you study chemistry, biology, but before it's just science. So I always been interested in science, and it's quite fun because my first test in chemistry in high school, I had a very low grade. and also, well, I really like challenges. I mean, when it happened to me, something like that is like, okay, I have to study more because I have to obtain a higher mark, because in the end I have to obtain a good result. And so then I really started working on chemistry, and everything went well. And then I studied biology. I was really, impressed by, what nature made. I mean, the equilibrium, the cells, thinking about everything, all these different parts that are interconnected, and everything that happens is due to a reason. And so I think it has also some, more spiritual meaning through science in some way. But yes, I mean, I studied history, geography, but I didn't enjoy it. Never. So it was also always about science.
Science, yeah. Yeah. So where did you go to university, and how did you end up in Spain for your postdoc?
Yeah, so I studied in Bari. Well, Bari is in Puglia. It's like, for you to visualize, is the heel of the boot, basically.
Okay.
And we have there, it's a quite big city, and it's very active from different point of view. From a cultural point of view, there are several activities, theater, concerts, and I have to say that was also very, very good for me to be surrounded, from this. And then we have different universities, very good universities. And I had I was lucky because I have these very good professors also in the university. So I'm quite happy, regarding my studies in the university. I mean, there are sometimes, some exams or some professor you don't feel very well, from a relational point of view.
Yeah.
But I think it's also a challenge in that sense, because you have to understand how you can pass the exam. And I mean, I always like the university also, and studies in general, because can help you understanding how to face some problems or issues you will have in your life then. And yeah, so I studied there in Bari. I had to, decide which kind of degree program. So I was just checking these books where you have all the exams listed. And I found pharmacy. Well, in Italy, we have in pharmacy 2 different courses. One is pharmacy, that is more for people who want to work in a pharmacy. And then we have this pharmaceutical chemistry and technology. That is the one I made. That is more focused on, it's more for people who want to do research and development. And so I read the exams and I thought, yes, this is my university. And I was quite lucky also because in Italy, now I think not, but before you have to do a test, you have to pass an exam to go to the university. So this is the only one I did. And I really enjoyed all the exams, but I know I was very lucky because I know people that were not able to find, the degree program at the first time. And it's very difficult then to find your way also because you see people go ahead and you are not able to understand your way. So it's
but when you think about it, it's also a challenge. Maybe that's when these setbacks happens, as you mentioned, sometimes not having a good relationship with the professor, and then it's a challenge. You want to understand where do they come from and what can you learn from them?
Yeah. No, and also, the other thing that I really liked is that in Italy we do a lot of exams, oral exams. So every time you have to answer questions, in real time. And if we have, a written part, is always written and oral exam. And so I think that it really helped me to learn, to feel comfortable speaking in front of people, in front of audience. And it's something that it's very useful for me now. And I see also other people that did not do it in this way. I see some, problems in that. And I think it also helps you to be more confident in yourself also. And also, I think it helps you to be brave because you are not afraid about telling something that you think, because every time we are afraid about that what we are going to say is something that has no sense or it's something silly, for instance. But it's not true. In research, I think it's very important because every time you write a project, you are proposing something that does not exist, and you have to defend your idea. So I think I learned a lot of things in not just science, also because everyone knows that you will forget 90% of concepts.
Yes, it's a piece of paper at the end.
But you have to develop your tools to do what you want. And so I studied there and I did my experimental thesis in medicinal chemistry. And it was my first, how you call it, haha moment.
Yes.
Because I had 3 options that I was thinking about. So, technology, so formulation, then organic chemistry and medicinal chemistry. And they were all great professors, nice labs, but in the end I went in the medicinal chemistry one. And it was all, connected then, because I then did my PhD there in the same group. I was not sure to be able to do the PhD because we have there, like a number of positions open per year. You have to do an exam. And there were several people and a few positions, but I was able to have it. And then during my PhD, I did this 1 year in Santiago, where I went because, Professor Edi Sotelo, who was my tutor during this PhD visiting period, and then for my postdoc, and now is the scientific advisor of Celtarys. He had been in Bari for a postdoc 10 years ago. And so this was the connection. So basically, since I always think that when I decided to do my experimental thesis in medicinal chemistry, everything started in this path because it was all connected.
Yeah. That is wonderful. And it's interesting because in different segments of the podcast, we always talk about advice to scientists. But there's also the aha moments that you mentioned. And I think one key thing that you just told us about is how important networking is, how important it is to go out, one, to put yourself out there, and to trust yourself to say, this is my idea, these are the reasons why I think I'm proposing, or why I'm proposing this project. And you never know who you meet and where you meet them.
Yeah.
And in order for you to move forward in your career, you need to put yourself out there.
No, I completely agree. And another thing that I would like to add to this is, I mean, I always did in this way. I don't know if it is correct or not, but it's also that when you have to choose or give an answer about something that is important, for your career, I don't like to think about it too much. I mean, because you will always find, I mean, you can do this, conventional list of cons and pros, but I would not spend too much time on this, because in the end, as a lot of friends of mine told me also, you already know what you want to do in your So I think that, for instance, for Santiago, my professor came back. He told me, I've just been in Santiago. They have great labs, because there are, different research centers that are very, about excellence in research. They have amazing laboratories, resources, very expert researchers. And he told me, do you want to go there? And I said, okay. Because, do you want to go in Galicia? And I told him, where is Galicia? and it was great. I mean, I said, okay, let's go. Because, if he told me like this, I was very, I know that I could, it was very giving me good advices as a supervisor. And then I was very happy about that. So I found a very nice place, very nice people. It was a great experience.
Yeah, and absolutely. And it led you to Celtarys and being the CSO of Celtarys today. So it's a small world. You were mentioning about the pros and cons in the list. So to one movie reference I have that popped into my mind as you were mentioning it. Another thing is that I always tell people, close your eyes and put yourself in situation A versus situation B, and try and figure out which one makes you happier.
Yeah.
But then my movie reference is the last Matrix movie, where when they say choice is an illusion, you already know what you have to do. And when you were talking, I was like, wow, I love this quote. I was like, wow, this is actually quite cool. Choice is an illusion. You already know what you have to do.
No, I completely agree. I think that the problem is that sometimes we think about what is the good thing to do.
Yeah. Or maybe I think, I don't know if it's an academic thing of what would my mentor do, or what would the community find acceptable in a sense. And I'll add another layer to it, and you tell me if you agree or not. It's the female thing. I think we, as women, and maybe I'll talk about myself, especially young, when you're doing your PhD or your master's, you're a little bit unsure. You haven't built that confidence yet. Therefore, you're looking for the answer outside of yourself. Maybe men have the same situation. I think my husband would say men have the same problem, but I think we live it differently.
Yeah, I think we live it differently, yes.
And then with time, you realize, no, choice was an illusion. I already know what I want.
Yeah, no, I completely agree. And you have also to be a bit lucky, because you are not sure that the decision you are taking will be, in the end, you will be happy in that, because you cannot, oversee the future.
No, that's true. I think it's also about, I think any choices that you make, and we're getting very philosophical here, but I like it. Any choice you make is going to lead you to a route or to a road that you need to take in order for you to grow, to learn something and then move. And it's not a bad choice, it's a detour. And it might be a painful detour, but it's a detour.
No, indeed, for example, I think that also discussing again about the fact of go outside your comfort zone in some way, no? I think that everyone in the world, I mean, scientists, not science, anyone should live like 1 year abroad.
Yes, I agree.
Because you really learn how to relate to people from different culture, from different point of view, and they help you to see yourself and your life from a different point of view. And so you can do like a puzzle. So you can take from each experience what you like more and then build your personality in the end. No? And I think, for me, it has been especially about scientific personality.
Yes.
Because from the from the protocol to do an amide conjugation reaction to I don't know how to do a purification column, but also how to manage a team, because you have different ways in which it can be done, and you see live how it works and what you like the most. So for instance, now that I'm a team leader, so I have 6 researchers in my team, I have to learn a lot of things about leadership and management, but I'm basing now on what I saw before. And so the kind of management, so that kind of leadership, I consider that is the best, and that I liked as a team member.
Yeah.
And so it helps you to, because then you will find you will have to face different new experiences, and so you can base on what you experienced before in some way.
No, I agree, and I think and sometimes people are lucky, like you seem to have been, where you had the mentors and the leadership that made you, like you had that good mentoring relationship where you said, oh, I like how I was mentored, therefore I am going to continue mentoring. But the opposite can also be oh, I didn't like how I was mentored, therefore I'm going to be different.
Yeah,
both of those are fine.
Yeah, I agree.
So let's get back a little bit to GPCRs. Where do GPCRs enter your life at this point?
Well, in Santiago de Compostela, actually. Yeah, because the lab of Professor Sotelo, they have been working for the last 20 years in GPCRs modulators development. They work on the medicinal chemistry, and they collaborate with the group of Professor Majellaro, also in Santiago. Professor Ed is located in the SICUS, that is more on chemistry, and then there is like 10 minutes walking CIMUS that is focused on the pharmacology. And in CIMUS they have this high throughput screening platform called InnoPharma, that is one of the nodes of this network called EU Open Screen, and they do high throughput screening on GPCRs. So basically and also it was very nice for me when I went there for my PhD, because it was the first time that I did molecules, and I walked 10 minutes. Okay, can you evaluate this compound? Thank you. So it was, it's very efficient, because if you want to obtain a lot of info, especially because, for instance, we have been working a lot on antagonists of the adenosine receptor, the A2B, and now there is the crystal structure, but at the time there was no crystal structure. And so the Eddie works a lot with multi-component reactions, so basically generating these modulators using these reactions in which through a one-pot reaction you have build a scaffold and changing, you can change the decoration. And it was very important, for instance, in the case of the A2B, because we could collect a lot of information, and then also developing computational models with Hugo Guterres, that is another researcher working on computational chemistry in the GPCR field. And we identified an enantiospecific interaction with the A2B. So it's basically the same molecule having a chiral center, just changing the enantiomer, R or S. One was inactive and the other showed an high affinity.
Wow.
And so it was very interesting. And I mean, now you have the crystal structure, but even though you have the crystal structure, I think that there is a lot of information that you are missing. So I don't know, maybe it could be, a quiet conventional point of view from my side, but I think that it's important to have experimental data. Several experimental data.
The structure is a snapshot in time.
Mm-hmm.
And that's stabilized. And structure often requires modifying the receptor, stabilizing the receptor. And you don't know if that particular structure, that particular pose, has any activity associated with it, or any physiologically relevant activity associated with it. But I think it's a beautiful example of, having one enantiomer inactive and the other one fully active. I think it's just, this makes the world make sense. When you know that the positioning of the molecule within the binding pocket and the interactions that molecule that enantiomer makes with the different amino acids in the GPCR is key. We don't have that map, and having a structure doesn't necessarily lead you to that particular
No, I agree. So it was very, very, and we have been working also on other GPCRs. Well, I worked also with the CB1 and CB2. Well, actually, I've been working on, when it was my previous academic life, on new modulators of CB1. We were trying to avoid, adverse effect, so related to the activity in the central nervous system. And so we tried to develop compounds trying to block, the permeability of the blood-brain barrier. And also been working on 5-HT2A receptors, so developing some analogs of pivamanserine, always using multi-component reactions. So yeah, it was very, very interesting. And again, it was very nice because you have already a feedback. Indeed, this is also something that helped me and helps me a lot now when I start my own projects, because the first thing I do is, okay, I want to do this molecule. How do I evaluate the biological activity? Because in the end, you will have a lot of molecules and you don't know. So maybe they are very good, but you don't have the biological data. You don't know how to evaluate it. And, it's a pity because, so I think you should. And this is why also I think that the Celtarys is very important in that sense. I mean, what we can really, how we can support, the research and development activities, because everything starts from the biological assay for your target.
Yeah.
And this is what we want to do, because, in the market, there are several fluorescent ligands, but developing a new fluorescent ligand is complicated. It's not easy, because you have to put the linker, the fluorophore, find the best linker, find the best fluoro. So it can take quite time. And the advantage we have is our technology, because we can do it convergently, efficiently, in a short time, customize. I mean, just for the application you want, for the property that is important for you, because everyone knows that affinity and functional activity is not the same. So are you interested in developing a binding assay? So I will focus on affinity. In the case of functional activity, I will try to keep the functional activity.
Yeah.
And I mean, every time we have new targets, so even though you have a ligand, it's difficult that you can find it on the market all the time. So
yeah. No, to be honest, being on the drug discovery side of things, when I initially thought about fluorescent ligands, it was, oh my God, this is going to be so difficult to develop. It's going to take time. It's going to cost money, and there's no guarantee that it will work out. So, and to your point, with your technology, these are the blockers that you're getting rid of on the market. But before we dive a little bit more into what does Celtarys do, how did the company come to be?
Yeah, so basically the technology of Celtarys was the PhD thesis of Dr. John Yatsuache. He was a PhD student in the group of Professor of Adi Sotelo, and he could not publish anything about it, because when you see that what you are working on as a commercial interest, if you want to patent something, the first point is that there hasn't been no example before your application.
Yeah.
So, Johnny is a great researcher, and he had a lot of things that he could present and publish, but especially about this technology, he could not. And I think they started working on this. And then when in 2018, when I came back to Santiago, I started working on the validation of the technology and all the process of technology transfer from university to industry. And there in Galicia, there are several grants. And from this point of view, I was really impressed. They have grants that support you in this transfer of technology. So there are always new startups, biotechs. There is a big network of BioTech, Bioga, in Galicia. And so we had this 2 years grant in which they support, because we know a lot about science, researchers and scientists, but there is another aspect when you want to start with a company, other aspects that we are not, very comfortable with. So marketing studies, marketing plan,
business plan,
business plan, policy, how you want to manage the intellectual property, investors.
It's a whole other world. And, I'm glad we're talking about this, because it's not something that we get a chance to talk about a lot on the podcast. And there's a whole new world out there. You can have the best science. If you cannot communicate it, if you cannot, go through these steps of commercializing it, then it's unfortunately only going to be a publication. Yeah. And it's a pity. And you were lucky that you had this grant that allowed you to build upon that.
Yeah, because they really advised us about how doing different things. And so in the end of this grant, we had to constitute the company. And so in 2021, Celtarys was born. But yes, Celtarys, the history of Celtarys is also related to the group I mentioned you before, of Professor Majellaro, Jose Brea, the great pharmacologists who have been collaborating with Eddie for so many years, from in academy and industry, because they also participated in the development of different drugs. And so we have been experiencing as medicinal chemists that we wanted to substitute radioactivity, because all our compounds were tested by radioligand binding assays. And also discussing with Mabel and Jose, they told us that every time they were purchasing a new radioligand, it was like that the same amount really was good to perform less number of experiments. They were experiencing lower stability. And then, when you work with radioactivity, you are not democratizing science, because you really need facilities, training, you have to manage all this waste of radioligands. And so we had on this side this problem we were experiencing, on the other side our technology that basically allows to build these polyfunctional compounds. So we thought, why do not generate a fluorescent ligand? And so this is basically, and yes, so we constituted the company together with Mabel, and also the contribution of other researchers like Javier Sardina is another professor in organic chemistry in Santiago. And then we were also lucky to have very good investors then who trusted in us. And so now, I mean, we could start, and it was great because then we started to write projects. We are in different grants. In 2023, we granted a Pathfinder project about GPCRs with an amazing consortium. And I'm very happy about that because, I mean, the opportunity of working with such amazing researcher is something that does not happen every day.
Absolutely.
And so, yes, we did a lot of things now. So I mean, we are still doing our story, you know.
Yeah.
And also the aspect of, you know, at the beginning, it was me and the CEO, and I was the chief scientific officer and the principal scientist.
Yeah.
And now I have a great team of 6 researchers, both in biology, biologists, organic chemists. And so, yes. So nice.
Wonderful. Well, yeah, actually, you know, you were mentioning having the molecules and not having the biology to go with it. We're kind of two sides of the same coin. I'm coming at it from a biology perspective. You can have the best assays, but if you don't have molecules to test,
no,
what's the use? And you can have the best molecules, but if you don't have the assay to test it in, you know, a robust, reproducible assay with the right controls, then the molecules don't mean anything.
Yeah,
it goes both ways.
We need each other.
Yes, we do. Yes, we do. So where did the name Celtarys come from?
Well, actually, I have to say that the main idea was by Eddie, because when I have to be, I mean, creative in that sense about names, I have this, you know, blank mind. I cannot think anything.
You're a true scientist.
Yeah. Yeah. So, well, basically, we have been thinking about the main application of our technologies in fluorescent ligands. Even though, I mean, it's not just fluorescence because we work also with different readouts, but at the beginning it was the idea. And the grant we had, this 2 years grant, the project was called Fluoro Tools. Okay. And so we were thinking about something similar, but we were afraid that putting fluoro in the name, you know, make people think that we work with fluorinated compound. And so we have been thinking about that. And so also we have our motto, no, innovative chemistry to illuminate biology. So it was more about, you know, we want to shed light on, you know, research. And so we're thinking about, you know, light, stars, so the North Stars, Polaris. But we have been checking about Polaris, and there are a lot of companies named Polaris, also from different fields. So and basically it was, you know, a name that did not say a lot about us. Mm-hmm. So we wanted to give, you know, our sign in our name. Yes. And so Eddie proposed to add the root of Celta because Galicia has a Celtic heritage.
Okay.
And so we had this Celta, and so Celtarys. And the Y was basically because, you know, looking to the name, it looks more, you know, pleasing visually with this Y.
Yeah.
And so this is the story. And if you see indeed our logo, you will see the cross, the Celta cross in the middle.
That was going to be my next question about the logo, because I think it's a beautiful one. It's a very fascinating logo, which makes you wonder, where did that come from? Like, usually you do want a story behind it, like you just told us, because I think it just gives it more oomph, more value there.
Yeah, also because, I mean, we wanted to add, as I said, something about us that is not about me or about Eddie. I mean, Eddie's from Cuba, I'm Italian, we are not Galician, but I mean, we know that if we could do it, it's also because we have been supported by Galician community, Santiago, all, you know, the organizations that work in order that people can have access to these grants and develop their ideas in the end.
Yeah. That's wonderful. So the other question I had for you is, in general, who are Celtarys' customers? Like, what's that profile? I know we talked a little bit about, the science and how the company became, and about the fact that you also had to grow into the role and do things that were outside of the medical chemistry itself. But I'm assuming defining those funnels and defining who the customers were, who was the target audience, was also an exercise that you went through as a company. So if you can, anyone listening, in a nutshell, who are the customers? Who are you helping with this technology?
No, actually, this is an exercise that we had to do at the beginning. When I was mentioning we are good as scientists, but we don't know. So first exercise was about understanding who are our clients. I mean, not understanding because you know it, but it's not the same knowing it and thinking about it and write it and say, okay, this is my target. So basically, we support researchers in the preclinical phase, preclinical development of drug discovery. And we have different kind of customers. So from one side, we have pharma companies, big pharma, but also biotechs that work in drug development. Then we have CROs, so companies that offer services of screening, for instance, and they need to develop a new assay to offer this service about a new target. And then we work also with academic groups.
Interesting.
Indeed, we did different custom developments also, for instance, in US with the MD Anderson Center. And so basically we have been working with all these people, the researchers from these three main categories. And the needs are quite different because it depends on what you want to do. So for instance, for Professor Galcon, the MD Anderson, he needs to understand the effect of, the mechanism of action of, they identified a receptor that is similar to the MOR receptor, the mu-opioid receptor, fly. And so they wanted to see, they saw that they have some effect of, more agonists, but they wanted a more agonist to demonstrate the mechanism of action. Then we have, for instance, customers that are starting, as I mentioned before, a new research project. And so there is no fluorescent ligand available in the market. They want to develop their own assay, or those that are available are not suitable, because the fact that it's available doesn't mean that it's okay. It's good for your application. And so, for instance, we have like pharmacologists who tell us, please develop this fluorescent ligand. And so there we work and we identify the best ligands. So we ask about, do you prefer agonists, antagonists, affinity, all these questions. And we identify the scaffolds and we start labeling them, and we then test the compounds. But from the other side, we have, for instance, pharmas that they have their heat or lead compound. And they want to label it because they want to study it deeply, or we have to label a compound of their set of candidates. That is not the heat, but just to develop the assay because the target is not very well known, so there are not too much ligands. So yes, basically, and the same kind of categories also for our portfolio, because we have also a portfolio of fluorescent ligands we commercialized that basically was how we started, especially because we wanted people to test the probes that we developed to trust.
Of course.
And this is what happened, because several customers started to buy the reagents and then ask us for our customer development.
So. Yeah. You wanted them to taste the cake.
Yeah.
Or the focaccia, depending. No, absolutely. And that's how you build trust, because you and that's how you learn as well. Because in the beginning you were explaining, we were talking about fluorescent probes and linkers, and now you've kind of you're writing as you go the playbook of what customers need, and also what's missing in the field. And that I think is super valuable, even more valuable than having one product that you sell, because now you have a deep understanding of what's missing, and it allows Celtarys to then innovate. And then
yes, because I think that being before scientists, also from the, you really experience the what you think could help to make everything easier. But it's something that never stops also, because you have to continuously talk with clients, ask their needs, and try to focus your pipeline or main priorities, also basing on what you understand is the most important thing for them.
Absolutely. And it's interesting because when people are in academia, and maybe I'm talking 10, 15 years in the past, I think whenever you go outside of academia, you don't do science anymore. And that's not the case. You do if you're a scientist by heart, by genetically engineered to be a scientist, then no matter what role you take, you will still do science. Because it sounds to me, from what I'm understanding, is you're not only creating these probes, but you're actually working hand in hand with customers in order to understand their problems and find a way to help them.
Exactly.
And that involves science. You can't do it without it.
Yeah, also I really like my job because we always do research and development. It's as I always say, it's difficult that we prepare the same molecule 2 times. Because it's everything really customized, and I really enjoy it, because we have a conversation, because I like to describe us as your partners in assay development. So we want to solve issues related to assay development, not just fluorescent or fluorescent ligand, but indeed it happened several times. So we had some conversations. So I have this problem, how can you help me? And so we provide a technological solution. So in addition to our technology, it's not, our technology is a tool to obtain the tools, so the reagents basically. But the reagents have specific properties depending on the solution that we want to provide. So we start from the problem, we understand which can be the solution, we apply our technology to generate the reagents that will help to implement this solution in the assay. And it's very, I'm very gratified because there's a lot of gratification, because you really, when it works.
No, absolutely. But even when it doesn't, I feel like there's a lot of lessons learned then.
Yeah, no, because I have to say that most of the projects we did, majority of times we were able to solve the problem. Indeed, we have customer that work with us more than one time, and so this is our solution really helps them in the end to support their research activities. And so it's like every time writing a new project, learn a lot of things about targets, about problems, and so you always do research, basically.
It's like having multiple collaborators, where you come in and you try and work with them in order to solve a higher level question. And who's involved in the collaboration could be, as you mentioned, academics, could be CROs, could be biotech, could be pharma. But at the end of the day, it's the science. It's a high-level science questions that you're trying to solve with scientists from different disciplines, which I think is just wonderful.
And also the fact that you have that companies are trusting in you, in your experience, in your commitment.
Yes.
So because we try to transmit that we are really committed to the result, or to having the support for the development of the assay. And so it's nice, because that all these companies are trusting in you, and you were able to make them trust in you.
Yeah. I mean, you were just mentioning this, and the image that I had, since we were talking about baking cakes and focaccia, is a satisfaction where your focaccia turned out great, and you just take it out of the oven, and you have that incredible smell, and you're like, oh, this was actually good. And I feel like that's how the customers should feel, and that's how also you should feel as a company, where you're like, oh, we did this together,
And everyone wants a piece of your
of course, and wants to come back.
Exactly.
Wonderful. Maria, top 3 aha moments that shaped your trajectory. I think we kind of talked about them, but if you can summarize them for the audience, that would be really fantastic.
Yeah, well, I think that the first one was the when I was, I had this position of PhD, because I was not sure about doing PhD, neither about achieving, obtaining the position. Also because it was a very, difficult moment in my life, just that year. And so I was not really focused on that. And so I think that when I started doing this and I achieved it, was like,
I can do this.
Then another one was, because I don't know if I told you that Celtarys was born in 2021, so just pandemic.
Yes, I think we don't talk about it enough, but yes, 2019, end of 2019, 2021, 2022, that's pretty much pandemic.
We have been doing a lot of meetings online. I have to say that from one side that it was a good thing, because we had access to meeting people that normally you could not, because everyone was using, Teams, and so we had a meeting with a big company, and we have been discussing about a development we could do, and have my lucky shirt. Well, Eddie knows.
I have one too.
Yeah, it's like it's a cactus. There's several cactus on it. And when I dress this in different meetings and conferences, I used to dress with this shirt when I had these oral, talks, and every time it was great. So I thought it was my lucky shirt. Then I understood that I changed the shirt, and
everything was the same. But side story, whenever I had to give a presentation, I would always buy myself a new piece of clothing.
Yeah, okay.
And I would wear it. I was like, in my mind, okay, I like it, it's new, and this is, and I would wear it then only, like not before. Kind of first.
So I was with my lucky shirt, and we had a great feedback from them, and we started a really good collaboration. And it was another moment that I thought about, okay, Maria, because it corresponds to my shift, from lab to CSO, to leadership. And so I was already in the early stages that I was thinking about, okay, it was good in the lab. Will I be good also in this? And also, will I like doing, being outside the lab? And so, yes, this was another moment in which I was like very confident in myself. And yes, these are the two main moments that I remember. And yes, basically these two.
I love it. I know we also talked about this in the beginning of the episode, but any advice to aspiring, scientists who are in either on the medicinal chemistry side, or even computational or biology in the GPCR field, if they want to continue contributing to the field in a role similar to yours? Any advice? Anything you've learned?
I think that a very good advice is about being multidisciplinary.
Yes.
That is something that I mean, because my degree was based on that. I mean, I'm chemist, I'm, pharmaceutical chemist. Is like we know about chemist, but we didn't study organic chemistry 5 years. We know about chemist because we want to obtain a compound, we want to be able to design a compound. We know about biology. I can understand the results of a biological assay. I'm not good in doing the biological assay because I've never done it before. But I think that it's very important also. Something, for instance, that I miss is the computational side. I mean, I understand quite well, but I think I could learn a bit more about that, because I mean, it's very important that you can see the wall paint, let's say.
Yeah. And I think that's a challenge in general, not only in your case. I think especially in biotech and pharma, it's very difficult to have a holistic image or view of everything going on. And for that, I think there's tools that you can use, but there's also approaches that you can look. We're humans. We're not robots. We're not machine learning or computational, and computational work complements. I think I was going to say it's the third side of the coin, but the coin, maybe let's say it's the perimeter of the coin. Anything that's computational, machine learning allows you to look at the data and get insights that as a human you would not be able to get out of, hundreds of data.
Yes. No, but I mean, also, I mean, you cannot be able to understand everything, but if you have a friend that can help you with it.
Yes.
And we come back to networking. That is what we were mentioning before. I mean, networking is also important for that, because sometimes when I feel lost or something I do not understand, I send an email, please, I have this data, can you help me understanding this? Or so even though you are not very confident in something and comfortable in something, it's very useful to have collaborators, other researchers you can ask to about it.
Absolutely. And going back to the collaboration aspect, if it is important to collaborate, it is important to network. And to your point, the medicinal chemists learning how to speak to the computational people, how to speak to the biologists, and it goes into circles. I think that's what advances science to move forward, because quote unquote us biologists have a specific language, and we need to understand what the chemists need. What do you need to know in order to modify your molecules and then give them back to me so that I can run the assay and give you the results, so that you'll have the molecule and then you'll have the data, and next side by side, so that we can sit together and say, here is the curve and here is the molecule, and by adding this group here, this is how the activity changed. And I think it's this collaboration that makes it so powerful.
No, I completely agree. Completely. And I mean, if it's this communication is missing, projects I think do not work in the end.
I think so too. And I think it's about that balance of ingredients. I think I'm hungry because I'm talking about food more and more.
No, but as you said, you can have great biologists or chemists, but if they cannot communicate, you will not achieve your goal. So
goes back to your point that you had about professors at university, where the relationship might have not been as natural, and it was on you to understand, okay, how is that person working, and what is that person communicating to me? All right, Maria, I really thank you for your time. I really enjoyed our conversation. Don't go anywhere. We're going to let people wonder what we talk about after we stop recording. Thank you so much, Maria.
Thank you.
Before you go, a quick reminder. We've partnered with Celtarys Research to spotlight innovative approaches in fluorescent ligand development and GPCR assay support. To learn more about how Celtarys can support your work, click the link in the description. If you enjoyed this episode, hit the like button. And if you're enjoying the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribed yet. One click helps us a lot. Learn more about Dr. GPCR in the description. Find all our fantastic features and everything you can benefit from becoming a premium member in the description. While you're on it, check out the Dr. GPCR University courses and share them with your network, friends, and colleagues. We also want to hear from you. Is there anything missing from your ecosystem? Can we make it better? Email us at hello@drgpcr.org. Thank you for joining us and listening to this Dr. GPCR podcast episode. A big thank you to our team, Yamina, Attila, Mark, Ivana, and Andreina, for making this possible. Until next time, stay safe.